And Daniel Husen on Tanglegrams for Reticulogenetic trees and network. So as probably you already know, since you are here in this conference, reticulogenetic trees is defined as a direct graph with a null cycle, and we say that there is one node with in degree 0, so no parents, that we normally call the root, and the leaves of these reticulogenetic trees are associated to a set of taxa that we are interested in. But this kind of representation is not suitable for what we normally call reticulate events, such as, for example, horizontal gene transfer, recombination, and hybridization. And to represent this kind of event, we normally use a structure that is called a reticulogenetic network that, like reticulogenetic trees, can have some cycle, even if only indirect cycles. So direct cycles are not permitted, but the fact that we can have indirect cycle make it possible to use this kind of representation to represent this reticulate event where the genetic material of one species comes from several species, just not one. So the fact that we can have more than one parent. So this is the... So as you well probably know, in systematic biology we have often we face the problem of comparing several phylogenetic trees, and one way of doing that is for sure using like distance measurement, like bipartition distance, triplet distance, and so on. But sometimes biologists, they have the need to have a nice drawing to see really which part of the trees are similar and which are not similar. So people started using Tanglegrams that are defined as a drawing of two rooted phylogenetic trees facing one with the other, and with no edge that are intersecting in the two trees, and with some connectors, or also called auxiliary lines, that connect matching taxa. So these matching taxa can be, for example, the same taxa in both trees if we are doing a multi-gene analysis, or for example, in the case of a co-speciation study, we can have in one tree the host and then the tree the parasite, and we want to match them. So this problem is an easy problem, so it can be solved in polynomial time if one layout is fixed. But if both trees can vary, that in this case is an MPR problem, and there are some heuristics that work pretty well for some cases. So what we want to do here is to give a definition for Tanglegrams for reticulogenetic networks and give the first algorithm for doing that. So why we cannot just use the definition for trees, but because the reticulogenetic network can be non-planar. So sometimes we are forced to have some edge that have to cross to have a drawing. So this is why we cannot use this definition. We have to give a definition that can have some edge crossing, but at the same time we want a definition that when we restrict our input to trees is the same definition that we already have for trees. So this is what I'm going to do in the next 10 minutes. I will give you so many definitions, like a bombarding of definition, but I promise to the next speaker you will not be all asleep, so please, one or two people has to decide and stay asleep for the next speaker. So I will try to not to make you sleeping, but we will see. So first of all, we have to know what is the topological embedding of a phylogenetic network. So it's simply a map that associates each node of the phylogenetic network to an order list of its children. So for example, here I have a drawing. So each drawing implies topological embedding. This is obvious. And for example, here, if I'm interested in the map of this node, I will put in the order list, in the order list, this one before, and then the second one. And if I'm interested in this one, then I will put before the leaf called B, and then the leaf called C. So this is easy to understand. Then I will use several times the concept of reticulate edges. So a reticulate node is simply a node with more than one parent. And otherwise I call it a tree node. So for example, the one in violet here it's a reticulate node, and this leaf here is, for example, a tree node. And then the edges that enter in a reticulate node are called reticulate edges, so the one in gray, they're all reticulate edges, otherwise I call them tree edges. So as you can see in this example, when you remove all the tree edges from the reticulate edges from phylogenetic network, you get a forest of trees. So the one that are on the right. So I will call that the Fn if the network is called N. And when I talk about the topological embedding of a forest Fn, it's just a topological embedding of all trees. So I tell you exactly how they have to be ordered, the node in each tree, but I don't tell you how the trees are ordered with respect one to the other. Okay, so this is a topological embedding. So this is, I think, the most important definition of this talk. So it's the concept of a non-interleaving order. So on the right here, there is a topological embedding. So it is the same as before. It's tau. So if I have a total ordering of the taxon set, I call it pi. I say that this ordering is non-interleaving. If, okay, here this is the formal definition, but I will just try to explain it for the biologists present. The idea is that if you have two taxa that are in the same tree, then if I want a topological, a total order to respect my embedding, but if my tree say that A is before B, then I have to listen to my topological embedding and say, okay, A is before B. So for example, here we have that A is before D because my first tree of Fn tell me that. So if I start my total ordering with D and then I put A, this is not correct. So this is the first condition. The second condition is that if we have like taxa that can be part of different taxon set of my forest Fn. So in this case, what is written in the second condition is that the two taxon set, they can be like one near the other in the order, or they can be one inside the other, but they cannot interleave. So this is what I'm saying in the second condition. So I put an example. So here I have, for example, the first thing is a non-interleaving ordering, so it's okay. So I have A, D, F, G, H. So then I put all my second taxon sets. So A, J, K. Then I continue with L, then B, C, E. Okay? So in this case it's correct because this part is inside here. And the second one is not correct because I have A, D, F, J, H. Then I put a part of the second tree taxon set. Then I get back to L, and then I go to K, and I continue. So in this case, I have an interleaving ordering. So now you tell me, okay, really nice. You give this definition, but why? This is why, because this non-interleaving property ensures us that it exists a drawing that avoid me to have tree edge crossing. So if you remember what I started at the beginning saying that a tanglegram for trees was defined with no edge crossing. So this is why I need a non-interleaving property, because if I just have I don't have any reticulate edges, so I am in the case of tree. The fact that I have a non-interleaving property then gives me the possibility not have any edge crossing. So the definition that I will give you in the next slide, when I am interested in tree, is the same that was in the literature before. And we all know how it is important to continue with the good definition before and not changing them. So this is just the formal definition. You don't have to be afraid. So the only thing I'm telling you is that I have two networks. They can be in different taxon set. They don't have to be the same taxon set. And so what I want to give you, and this is the definition of Tanglegrams, is a topological embedding for the two forests, F and 1. They come from the N1, the first network, and F and 2, they come from the second network, N2. And two non-interleaving total ordering of X1 with respect to my topological embedding tau 1 from F1, and pi 2 with respect to the topological embedding tau 2. So it's just what I'm telling you is that this is the way that normally we give a tanglegram. So this is one topological embedding, this is the second one, and this is the two in non-interleaving total ordering. And then, so this is the last definition, so definition of an optimal tanglegram. So what we want is to have a tanglegram that minimizes the connector crossing. So this matching these lines that connect matching taxa. So we want to minimize this number, and among them, the one that minimizes this crossing number, we want to take the one that minimizes the interior crossing number. So the crossing number that we have inside the two networks. So here I give you 3 examples. So the first example, this is not a tanglegram, because the two, here we have 2 edge crossing, but these are 3 edges. So this is not a tanglegram because you have 2 3 edge crossing. The second is a tanglegram, because if you can see, the crossing, they involve only reticulate edges. But it's not an optimal one. Why? Because the third one is the same. They are the same 2 networks. One is a tree, but they are still network. And as you can see here, the crossing inside the first network is 0, and in the other was 2. So this is an optimal tanglegram, the one in green. Okay? So now I'm going to explain you how our algorithm works. So I will try not to be too prolix. So I think you know what is a split. So a split on a taxon set is just a bipartition of the taxon set. You normally use like for it's like it is a bipartition. Like the Robinson full distance is bipartition. And we use the term circular to define a class of split that are really interesting when you work on phylogenetic network. So this is, again, is the formal definition, but it's easy to understand the idea. So the set of split is called circular if you can put all the taxon on a circle, and then each split is just an interval on this on this ordering. So we call normally this ordering circular ordering of the taxon, and so in this case I just draw the line for FG and FISG. So this is the first split, is just this line. And these are really nice scala cell split because they give you a planar graph. So, but now it's not really important for this presentation. But if you use splits tree, normally you can get this kind of stuff. So from a set of split, then you can get a matrix. So this is also something that people do. They take a distance matrix and they construct phylogenetic networks. So and we can go in the other way around. We can take a set of split that are weighted, and we can get a distance. So how we do that? We just for each pair of taxa x and y, we just take all the split that separate x and y. Means the x is in one part of the partition and y is in the other part, and we just sum them. And in this way we get a split distance from a bunch of splits. And we say that a distance matrix is called circular if we have a bunch of splits that are weighted, such that when we compute the split distance from the split, then we can get exactly the distance we wanted. So it's just a distance is circular, can be completely described by a bunch of splits that are circular. So I probably you know NeighborNet is one of the most used algorithm for computing phylogenetic networks, and this algorithm give you from a distance matrix a total ordering of the taxa that is circular with respect to the split. So it gives you a bunch of split and an ordering of the taxa. And this is the most important thing of NeighborNet is that it is consistent. So if you have a circular matrix, then it will give you the good set of splits. And this is something that we will use in the algorithm. So this is the last definition before finally giving you the algorithm. And then I will not bother you anymore. So a cluster, given a phylogenetic network, there are two ways of giving a bunch of cluster from it. What I'm giving you is what is normally people call hardwired cluster set. So it's just really as it is for a phylogenetic trees. You get the bunch of cluster that are, you just take the edge, so for example E1, and you take just the taxon that are below the edges, like in the tree. And then you can associate a cluster, a split to this cluster, taking the cluster versus what is not in the cluster. So, and we call this SN. So I really have to run now, so because we don't have so much time. So our approach is really easy. So we start with two phylogenetic network. We add a dummy leaf to both. It's called row. Then we compute this distance. So we take the split I told you, and then we compute the distance from the splits, summing up these two guys. Then we apply this neighbor net from this distance, and we get this circular ordering. This is why we use neighbor net, and we break this circular ordering into the dummy taxa, and we get a linear ordering that is called pi. So thanks to this linear ordering, we're going to order our two networks. So now I'm going to give you the algorithm for N1, but it's the same for N2. So just like really informally, you just try to find the topological embedding of the forest that is as much in accord with the linear ordering pi that I just computed with the neighbor net. Then we take all this topological embedding for each tree inside, and this is what we call tau 1. And then we find the non-interleaving ordering that is again minimizing this number that I don't have the time to explain is that, but again we are trying to be as much as accord with pi. So this is our approach, and this approach has the beauty of finding a solution with zero crossing when this solution exists. This is why we use neighbor net. We use neighbor net because this fact that if there is a zero crossing solution, there is something that is planar. But I don't have to explain. I don't have time. But there is an underlying structure that is planar, and thanks to NeighborNet, we have the possibility to finding this zero crossing solution. So then there is a problem how to actually draw this tanglegram, but no time. I'm already super late, so I don't have time to actually explain you. But it's also another fascinating problem how actually to draw a tanglegram in a nice way. Because if I give you a biologist lots of ugly drawing, people are not going to use it. So we also did the simulation. So we did it for binary trees because there are lots of software that work for binary trees, and so it's working pretty well. So I show you the simulation for. So this is the two method, and this is our method because we can also improve our method, and I show you an example in which we are even not really good, but this is because here the crossing number is 0. So the first one worked pretty well, but when the crossing number is not 0, this is not working really well. So we are even better in other picture, but I always prefer to show the picture we are not so good, like that people don't say, but in other cases you may be not so good. So this is the worst one we have in the paper. And then we also did it for binary networks. So yeah, this is just like we compare to each other, fantastic, we are good, because there is no software, no definition that works for network. So this is just like some different implementation of our software. And okay, here sometimes it gets really bad. There's like 1-2 instances that it can be really bad. But you have to think that if I have like 180 leaves, already if I misplace the one, I get like a crossing of 180. So these numbers are high, but if you look actually to the drawing, they're not really bad. Okay, so I don't have time, but we also apply that to a biological example, and we took like, oh my God, I have to go. Okay, no, don't worry. So I got also a fellowship, a travel fellowship. I was like shocked because I didn't know that it was a sound. So I thank people that paid partially my my travel here at the University of Tübingen, where I'm working. So thank you for your attention.